Hello everyone. In this session, I am going to discuss tissue nematodes. So let's start it. So basically here I will discuss two important tissue nematodes, that is Trichinella spiralis and D.medinensis. So as usual, I will be discussing the tissue nematodes under the standard headings of entire parasitology. So first in the introduction, I will let you know their other names, their discovery, then their morphological forms, their mode of transmission, the infective form, their life cycle, clinical features, lab diagnosis, treatment, the drug of choice, and the prevention. So we will discuss both these worms, the important forms under these headings. So let's start it. Before starting the individual worm, have a look on the classification of the nematodes, that is roundworms, based on their habitat. So based on their habitat, we divide the nematodes under two categories. The nematodes which resides in intestine are known as intestinal nematodes, and the nematodes which reside in tissue are known as tissue or somatic nematodes. Now in the intestine, there are small intestinal nematodes. You can see the 4 names, and there are large intestinal nematodes. You can see the 2 names. So there is a separate video on small intestinal, large intestinal nematodes. I hope you have watched that. If you have not watched, you can watch the intestinal nematodes. Separate video is available. Now coming on the tissue or somatic nematodes. In the tissue or somatic nematodes, 3 important parasites are there. The first is Trichinella spiralis, D.medinensis, and filarial nematodes. So filarial nematodes is covered in a separate video. In this session, I am going to cover these 2 in detail, that is Trichinella spiralis and D.medinensis. So I request all my dear students to take out your notebooks, take out your pen and start filling this table, the comparative table, while watching the video only. So we will start with Trichinella spiralis first. Let's start it. So coming on the introduction of Trichinella spiralis. The other name of Trichinella spiralis is Trichina worm. Why it is known as Trichinella? Why it is known as spiralis? Why this nomenclature? The meaning of the Trichinella is very minute, since it is like a hair of the human, single hair. It is like a thread, like very hair-like, very minute in size, number 1. And its larva is spirally coiled. I will show you the diagram. That's why known as spiralis, because the larva is spirally coiled inside the muscle. It resides in the muscle of the patient. It resides inside the skeletal muscle of the patient. So that's why it is known as Trichinella spiralis. Trichinella means very minute like hair, and spiralis because the larva is like a spiral inside the muscle. So that is the introduction. Coming on the morphological forms. 2 morphological forms are there, the adult worm and larva. There is no egg. So in the adult worm, there is male, there is female. Male and female fertilize. The female become gravid, and after becoming gravid, female directly release the larva. There is no egg stage. There is no egg stage. So this type of laying larva directly, it is known as viviparous. So this worm is viviparous. It is not oviparous. It is not ovoviviparous. So it is viviparous type of nematode. Okay. Now coming on the adult worm and the larva, the details. So adult worm, it is the smallest nematode. The male and female are very small. It is the smallest nematode among all nematodes. And as I told you, it is viviparous. Viviparous means the female directly release the larva without egg in the intestine of the human. Now coming on the larva, there are 4 stages in larva, L1, L2, L3, L4. Among them, L1 is infective for humans. L1 resides in the muscle of the human, the skeletal muscle, and inside the skeletal muscle, this L1 will form a cyst. And inside that cyst, the larva will remain in the form of a coiled appearance. That's why it is known as spiralis. Can you appreciate the coiled appearance? The coiled appearance is there. So this coiled appearance is present in the muscle of the human. It is L1 larva. And that's why it is known as Trichinella, means it is very minute, and spiralis means because the larva is like coiled inside the muscle. Now what is the mode of transmission? The mode of transmission is ingestion of pork, uncooked or raw pork or other animal, the muscle of which contains L1 larva. So when someone eat pig or pork, so the pig or pork muscle is contaminated with L1 larva. Right. So infective stage is L1 larva. L1 larva. Now in the life cycle, there is only one host. The one host is either pig or other carnivorous animal. The man is a dead host. It is an accidental host. Man is neither the primary host nor the secondary host, neither the definitive host nor the intermediate host. The man here is an accidental or a dead host. There is no intermediate host. Only one host. The definitive host is an animal. It can be pig or any other carnivorous animal. So let me tell you the life cycle in man, which is a dead end. which is a dead end or accidental host. So man acquire infection by ingestion of the pork, contaminated pork. So the muscle of the pork contains L1 larva. Man is ingesting it. So the L1 larva will go in the stomach of the man. In the stomach of the man, the L1 larva will be released because of the digestive juices. The L1 larva will go to the duodenum. It will go to the jejunum. From duodenum and jejunum, it will mature in the intestine into adult form. So L1 will convert to L2, L2 convert to L3, L3 convert to L4, and it will become mature adult, the male and female. The male and female become sexually mature. They fertilize, and immediately after fertilization, the male dies. Now the female become gravid. So female become gravid. She directly releases the larva in the intestine of human. I will draw the diagram also. The larva will go in circulation. From the circulation, larva will go in all the organs. Of course, the blood go to all organs, but larva can survive only in skeletal muscle. In all other organs, the larva will die. So it will survive only in skeletal muscle, and it will form a cyst there and remain in the form of a coiled form. And this is the dead end. And if it was an animal, another carnivorous animal may be eating this animal, so the life cycle continues. The life cycle can continue if it was an animal, but no other animal can eat man. Man eat animals. Animals don't eat man, right? So that's why it is a dead end in man. You got it? So let me draw the diagram for you. Let me draw a rough diagram for you. So I'm trying to draw a human being. I am trying to draw the digestive tract of human being. Okay, let's start. So this man is eating the pork, the uncooked or raw pork containing L1 larva. So infective stage is L1 larva. The L1 larva is going inside. It is going in the mouth, esophagus. It is going in the stomach. In the stomach, the L1 larva is released from the muscle. This L1 larva will go in the intestine. L1 will convert to L2, L3, L4, and finally the adult worm is formed in the intestine. The male adult and the female adult, they fertilize with each other in the intestine of human. So when they do the fertilization, immediately after fertilization, the male dies. Only gravid female is there. The gravid female lay the larva directly. There is no egg stage. So the gravid female lay the L1 larva. This L1 larva will go in circulation. This is the blood vessel of the human being. This L1 larva from the intestine, it will move in the circulation. From the circulation, it will go in all organs, but it dies in all organs except skeletal muscle. In the skeletal muscle, this is the skeletal muscle. So this L1 larva will enter inside the skeletal muscle. It will form a cyst there, and inside that cyst, it will remain in the form of a spirally coiled. So this is the dead end. Now no other animal is eating the man. If it was not a human, if it was any animal, another carnivorous animal eating this animal, so the life cycle continues. But here it is a dead end. Here in human it is a dead end. So you got the life cycle. Infective stage is L1. The mode of transmission is eating pork, and man is the dead end. Right? So this is the complete life cycle. The same flowchart is in front of you. Coming on the clinical features. It causes a disease known as trichinellosis. Trichinellosis having 3 stages. Here you can understand now. First, it is going in intestine. So that is enteric stage. After intestine, the larva, the adult forms are formed. The adult form fertilize with each other and larva is laid. The larva will go in the blood. In the blood, it is parenteral stage. And after going in the blood, the larva will go in the muscle. That is muscle encystment stage. You got it? So first, it is going in intestine. First, it is going in intestine, after that it is going in blood, and after that it is going in the skeletal muscle. So we are talking about the worm. So worm is first present, the male and the female are present in the intestine, that is enteric phase. Then they fertilize and lay the larva. Larva go in the blood, that is parenteral stage. Now the larva will go in the muscle, that is muscle encystment stage. So these are the 3 stages: enteric stage, parenteral stage, and muscle encystment stage. You got it? So in the enteric stage, parenteral stage and muscle encystment stage. In the enteric stage, patient have diarrhea, abdominal pain, constipation, nausea, vomiting related to the intestine. In the parenteral stage, when it is present in blood, there is hypersensitivity reaction. The patient can have fever, eosinophilia, periorbital and facial edema, and sphincter hemorrhages inside the conjunctiva, retina, and null beads. So that is due to hypersensitivity reaction. And finally, the third stage is the muscle encystment. So it will go and reside inside the muscle. So the patient have muscle pain, that is myalgia, muscle inflammation, that is myositis, muscle edema and weakness. Which is the most common human body skeletal muscle which is involved? The most common muscle is extraocular muscles of the eye, followed by biceps. So first is extraocular muscles of the eye, followed by biceps. Learn these two muscles. After that, jaw, neck, lower back, diaphragm, other muscles are also involved. Now coming on the lab diagnosis, what you will do? The best is to do the muscle biopsy. Either do extraocular muscle biopsy or bicep deltoid, this biopsy. In the biopsy, you will find the larva. Can you see? It can be a spotter. It can be a IBQ in your exam. Can you see here? You can see inside the skeletal muscle multiple cysts are present. And inside the cyst, here also you can see the multiple cysts are present. Inside the cyst, you can see the spiral larva, typical spiral larva, coiled larva. So it is typical L1 larva of the Trichinella spiralis. You can take the blood sample, look for antibody. You can take the blood sample, look for antigen. You can do skin test. The skin test here is Batchman test. It's a skin test. It is delayed hypersensitivity. You can do the radiology. In the radiology of the muscle, you can do the X-ray of the muscle. Inside the X-ray of the muscle, the calcified cyst can be seen containing the larva. Or in the blood, non-specific is eosinophilia. So the best investigation is the muscle biopsy. It's a PYQ, repeated PYQ for Trichinella spiralis. Now coming on the treatment, the drug of choice is mebendazole. That's it, right? The prophylaxis, properly cooking the meat, the pig meat, the pork, or freezing it. Properly cooking it and freezing. If there is a delay in cooking, it should be freezed at -20 for 3 days. And smoking and salting and drying may kill the larva. So that is the prophylaxis. So we are done with Trichinella spiralis. Let's solve some PYQs on it and move to the next worm, the next parasite, that is D.medinensis. So okay, we will solve the PYQs together. So let's directly move on. D.medinensis. So D.medinensis, what are the other names given to D.medinensis? It is guinea worm, dragon worm and serpent worm. Guinea worm, dragon worm, serpent worm. Please learn the other name. The other name of Trichinella spiralis was Trichinella only, right? And here it is guinea worm, dragon worm or serpent worm. Dragon, serpent is one and the same thing, right? India was declared free of dracunculosis in the year 2000. After year 2000, there is no disease. There is no disease for this of this parasite in India. So in India, it is eradicated. In India, not in the globe. In India, it is eradicated. So in 2000, India was declared free of dracunculosis medinensis. The adult form remain in subcutaneous tissue. Now see, Trichinella was remaining in the muscle, the skeletal muscle, and dracunculosis is adult form is remaining, it remains, the habitat is subcutaneous tissue. Now coming on the morphology, here also adult form and larva is there, no egg is there. So here also it's viviparous. The worm is viviparous. So adult male, female, they fertilize, the female become gravid and female directly release the larva without egg. Mode of transmission is drinking the fresh water from the stagnant sources, stagnant, not flowing, the stagnant sources. It can be a lake, it can be a pool. So drinking the water from the stagnant sources containing cyclopes, and inside the cyclopes L3 larva is present. So L3 larva is the infective stage. So the person is drinking the water contains the cyclope and cyclope contain the L3 larva of dracunculosis medinensis. So this cyclope containing L3 larva is the infective stage. So what is the infective stage? It's L3 larva, that is filariform larva. In contrast to Trichinella, where the infective form was L1 larva present in the meat of the pig. Here the infective form is L3 larva present inside the cyclope in the fresh water from a stagnant source. So here drinking is the mode of transmission. There in Trichinella, ingestion of the pork meat was the mode of transmission. Now coming on life cycle. So definitive host is man, but intermediate host is cyclope. Here we have two hosts. In contrast to Trichinella, where we have only one host, that is any of the carnivorous animal like pig. There is no intermediate host. Man is the accidental host there. Here we have man as definitive host and cyclopes. The scientific name of cyclops is copepods. So we can say it's copepods or cyclopes. Let me tell you the life cycle. Let me read it first, then I will draw it for you. So man acquire infection by drinking water which contains the cyclope, and inside the cyclope L3 larva is present. So cyclope will go inside the stomach. The cyclope will rupture in the stomach and L3 larva will come out. L3 larva will go in the intestine. From the intestine, it will penetrate and go in blood. Via blood, the L3 larva will go in thoracic muscle. Inside thoracic muscle, the L3 will convert to L4. L4 will convert into adult. So it takes place in thoracic muscle. After converting into adult, they fertilize there only in thoracic muscle, the male, the female. The female become gravid. The gravid female migrate from the thoracic muscle into the blood again, and via blood it will go to the skin. It will go to the subcutaneous portion of the skin, and especially that portion of the skin which can come in contact with water. So most commonly it will go to the foot. It will go to the leg and the foot, and there it will form the blister. It will form a small blister by secreting certain enzymes, and the adult female discharge the larva directly in water when the foot comes in contact with water. Let me show you. Can you see? So here the blister is formed. Can you appreciate? So when this come in contact with water, the female directly lay the larva in the water, not in human body. So the gravid female directly lay the larva in the water. You got it? And the larva will go in the water. L1 larva will go in the water. L1 larva is ingested by the cyclops. L1 convert to L2. L1 will present inside the cyclops and convert into L2, convert into L3 inside the cyclops only, and now another human being will ingest the water containing cyclops containing L3 larva. So in this way the life cycle continues. Let me draw it for you. So imagine this is a human being. Let me draw a human being for you. So this is the human being. Okay, this human being is drinking water, the fresh water containing cyclops having L3 larva. So L3 larva is the infective stage. So the cyclops containing L3 larva will go inside. So let me draw the digestive tract. It will go in the mouth, go in the esophagus, go in the stomach, and then go in the intestine, right? So this L3 larva going inside in the stomach, the cyclop will rupture and L3 larva will release. So L3 larva in the intestine it will go. In the intestine, it will penetrate the intestine. Let me draw the blood vessel. It will go in the blood vessel. Can you see? This is the blood vessel. So this L3 larva go in the blood vessel. Via blood vessel, it will go to the thoracic muscle. So let me draw the thoracic muscle. In the thoracic muscle, this L3 larva will convert into adult worm. Adult worm have male, have female. The male and female are present. They will fertilize, and the female will again come in the blood. The gravid female come in the blood. Via blood, the gravid female will go to the foot. It can go to any portion of the skin, but especially foot, which is having a high probability of coming in contact with water. So the gravid female come in the foot and form a blister there, a small hole there, and will remain there. As soon as it will come in contact with water, the gravid female will release the L1 larva in water. L1 larva will go in water. L1 will be ingested by the cyclop. Inside the cyclop, L1 convert to L2, L2 convert to L3. Now this cyclop is ingested by another human being. In this way, this life cycle continues. I hope you got the complete life cycle. The same is written in front of you. You can read it. So a blister is formed in the subcutaneous tissue. You can see this is the blister. So it can occur in any part of the body in the skin, but especially the part which is having high probability of coming in contact with water. So especially in the leg and the foot. Now coming on the clinical feature. In the clinical feature, it causes a disease. This disease is known as dracunculosis. In the dracunculosis, a papule is there. First, a papule is formed that will rupture and a blister will form. A blister is formed that is painful, from which the worm can emerge, right? And there is local erythema, urticaria, fever, nausea, pruritus is there. So this disease is known as dracunculosis. In the lab diagnosis, we can detect the adult worm, we can detect the larva. For detecting the adult worm, we can do the X-ray. If you do the X-ray, you can see the adult worm inside the skin in the subcutaneous tissue. For seeing the larva, just put cold water over the blister. If you have suspicion, just put cold water. Just put a cold water over this blister, the larva will be released. So artificially, you can release the larva, right? So on contact with the cold water, on the leg ulcer, you can see a number of motile larvae come out, right? You can take the blood sample and look for antibody, and in the blood eosinophilia, the non-specific can be there. Treatment, there is no drug to treat dracunculosis, no drug, no anti-helminthic drug. So you can remove the worm with the help of a stick. The female worm is present here now, just in the blister. You put a stick and take out the female worm. Can you see here? The physician is taking out the female worm. Can you see this thread-like female worm? It is taken out manually. So So worms extracted using a stick, and you can apply the pressure, heavy pressure to take out. Heavy pressure should be applied, right? And not heavy pressure, the moderate pressure, because if you apply the heavy pressure, it can lead to allergy also. The worm can break and it can lead to hypersensitivity reaction. Symptomatic treatment can be given, analgesics can be given, but no anti-helminthic drug is there to treat it. So that is the thing. Now I told you in India it is already eradicated in 2000. Actually in 1984 a national program was launched, National Guinea Worm Eradication Program. So since 1984 it took many years till 2000 to get eradicated from India. It was launched by the WHO. So in this program two things are done. The National Guinea Worm Eradication Program, two things are done. Number one, provision of the safe water which is free of cyclope. And number two, cyclope control is done with the help of a larvicide, that is abate. So because of this, it is eradicated from the India. We are done with the two worms, the two tissue nematodes or somatic nematodes, Trichinella-spiralis and D.medinensis. Let's solve some PYQs on it and we are done. So the first question is in front of you. Can you tell me the correct answer? Dragon worm or serpent worm is it Antrobius vermicularis? Is it Trichuris trichura? Is it D.medinensis or Taenia solium? Of course, the dragon or serpent worm is D.medinensis, right? The next question is in front of you. All of the following are true regarding D.medinensis except, they are asking except. So it is eliminated from India. It is true or false? I guess it is true. It is limited in the world only in some tropical and subtropical region, and only in limited regions it is present, right? So it is also true. No animal reservoir. No, it is also true. Yes. But see the option number B. Nirridazole prevent the transmission of the disease. I told you there is no treatment, no drug. Nor nirridazole, nor any mebendazole or any drug is not effective. So no anti-helminthic drug can prevent its transmission, can kill the worm. No. So this statement is wrong. That's why we will go with B, because in the question they are asking for except. Coming on the next question. Guinea worm was declared eradicated from the India in which year? 1999, 2000, 2001, 2002. So I told you in 1984 the government of India, the WHO launched a National Guinea Worm Eradication Program, and it took 16 years to get eradicated. In 2000 it was eradicated. So the correct answer here is 2000 because of this program. We are done. Let me revise it quickly, right? Can we revise it? Can you help me? So let me revise quickly. Trichinella-spiralis and D.medinensis, the two tissue or somatic nematodes. So here the other name is Trichinella only. Here the other name is dragon worm, serpent worm, right? You can learn the other names. Guinea worm. So the D.medinensis is eradicated from the India, not from the world. So it is eliminated. Better to use the word eliminated from India in 2000 because of the national program. In the morphology, here also we have adult and here also we have larva. Here also adult and larva, no egg stage, no egg stage. The mode of transmission is different. The mode of transmission here is eating pork containing L1 larva. Here the mode of transmission is drinking water. The water from the stagnant source containing cyclopes. The cyclopes contains the L3 larva. So infective form here is L1 larva and here is L3 larva, right? In the life cycle, here we have only one host, the definitive host, no intermediate host. The definitive host is any carnivorous animal like the pig, and man is the accidental host. But here we have both hosts, the definitive host and intermediate host in Dracunculosis. The definitive host here is the man because male and female are fertilizing in the man. The sexual intercourse is taking place inside the man, and intermediate host is the cyclopes, right? Clinical features I told you very clearly. Here the disease is trichinellosis. Here the disease is dracunculosis. Here in trichinellosis we have three phases: the enteric phase, the parenteral phase, and the muscle phase. The main thing is the muscle pain, muscle inflammation. And here in dracunculosis, on the foot or the leg, a ulcer, a papule, a blister is there. The lab diagnosis you can understand here. Main thing is muscle biopsy, and here we can look for the worm and the larva. Treatment here, drug of choice is mebendazole. Here there is no drug of choice. Manually the worm has to take out. That's it. So we are done. I hope you learned the topic well. Before ending, let me tell you something. You know what is the difference between ordinary and extraordinary? The difference between ordinary and extraordinary, a little bit extra. So if you want to become extraordinary in your life, right? So you have to do a extra effort, a little extra effort daily. If you daily do a little bit extra effort as compared to others, you will become extraordinary. Thank you. Wishing you all the best.